Okay, good evening, everybody. Welcome to our second. Get once I get the IT down. So again, welcome everybody, and thank you for coming out tonight. And thank you to those of you that are online for this second in our series. Much nicer night tonight than it was last week. It was a lot of rain and a lot of dreary, and it's a nice night tonight. So thank you for coming out. So just a reminder, this is our second in the series, and tonight you're going to hear from Dr. Jenny Neslage, who I will introduce in a minute, and she's going to tell us about Menhaden. But the following week, next Tuesday, April 16th, we're going to hear from Dr. Michael Gonzia and Andrew Hayes, who are some of our biogeochemists here. Their offices and lab spaces are actually in this building. And they're going to tell you a little bit about that new initiative I talked to you about last week, the anthropogenic changes in estuarine systems. And so this is a new initiative thanks to a large donation from Brian Hockheimer and Marjorie Wax. And this is for us to look at contaminants, particularly emerging contaminants here in the Patuxent River and also globally, to try to address some of these concerns on emerging pollutants. And then we're going to finish up on April 23rd with Dr. Chris Rao, who is actually the building opposite. He's one of our ecologists, he's a herpetologist, and does a lot of work with amphibians and reptiles. But he's going to talk to us specifically about how humans are impacting biodiversity. So I mentioned this last week, but there's flyers at the back and out in the foyer. Please take a look at our Chesapeake Dolphin Watch. This is an amazing program. It's been going on for 7 years, and it's thanks to community members like yourselves that are giving us this data. And it's a phone app, and so anytime you're out on the water or you can see a dolphin, share this widely with your friends and family. It gives us really great community data and tells us a lot about where dolphins are in the Chesapeake Bay and how many of them are. So for those that you weren't here last week, we're seeing them way further in the north now in the Chesapeake Bay, and it's thanks to this program that we're learning a little bit more about dolphins in the bay. And of course, I can't finish the introduction without asking for the support of our graduate students. We have a great education program here, as highlighted recently by our USM Chancellor and Vice Chancellor, so Jay Perman and Michelle Masusi. UMCES has a fantastic role in training our next generation of environmental scientists. And you only have to open the news and see there's another news story, there's another contaminant in the water, there's another environmental problem, climate change. And so this is what we're doing, where we're educating our next generation of environmental scientists that are going to be tasked providing solutions for these ever-increasing emerging environmental concerns. So there's a QR code there and also leaflets around that you can help support our graduate students. So with that, I would like to introduce Jenny Neslage, who is an associate research professor here at CPL. She's been here a number of years, I won't say how many, and she is one of our esteemed fishery scientists that are known internationally, globally for their area of expertise. Jenny specifically focuses on assessing fish and wildlife populations and addressing resource management issues. Remember, this is something that's very unique to UMCES. We have a state mandate that we provide advice to the state for things like fisheries management and other environmental concerns. So specifically, she focuses on topics such as population dynamics for a variety of different fish species. She's going to talk about Menhaden tonight, and a little bit on fishery stock assessment, and also the general fields of fishery and wildlife ecology, and also invasive species. So with that, please join me in welcoming Dr. Jenny Neslage. Thank you. Oh. The Zoom is seeing this, but I can't get to share my screen now. Hey, Mike, how do I get this so I can screen share? Give us a couple minutes technical change here. What are you doing, Alex?
Tom was the expert on this. Yeah, it's really weird. I knew this was going to happen as soon as we tried it. As soon as we switched presentations. Yeah, that's the thing.
Next time. Okay, I just drag it across.
No, screen share it from there, then drag the Zoom back.
Do I do the screen share now and then drag it?
Yeah.
And then it went away again. There it is. So it did it. It went back.
Yeah.
Thank you, Mike.
All right.
2024, and we're still figuring out. There we go. Does that work?
Great. Well, good evening, everyone, and thank you for coming out. I'm so excited to talk to you about my favorite fish tonight, Atlantic menhaden. As Karis mentioned, I'm a fisheries stock assessment scientist. And what that means is that I pretty much spend a lot of time gathering fisheries data, harvest data, and other scientific fish data, and try to figure out how many fish are in the sea and how many are safe to catch. That's essentially what I do. And keep going, okay. And I try to develop new methods for improving our understanding of fish stocks and how we determine what sustainable quotas are for harvested fish species. Let's see here if I can get this working. Yay. All right. So tonight, what I'd like to do is ask a few questions, or I guess I should say I'm going to try and answer a few questions. The first, and they kind of all have to do with my title. The first is, what is forage? What do fisheries scientists mean by forage? If you come from agriculture, it might mean something else, but we have a specific definition, and forage is really important when it comes to aquatic ecosystems and fish. And then I'm going to talk a little bit about what Atlantic menhaden are, if you don't already know them. They're a wonderful fish. And I'll talk a bit about where they can be found as well. In particular, focusing on our own backyard here in Chesapeake Bay, as well as the greater mid-Atlantic region, which I'm kind of defining here as New York down to kind of the Virginia border with North Carolina. And then I'm going to talk a little bit about when they're found in our own backyard and in the mid-Atlantic, because this has become a very hot issue. And a lot of the science has changed in the last 10 to 15 years. So this is new stuff that's been coming out primarily out of Chesapeake Biological Lab. So I thought you'd be interested to know more about it. I'll talk a little bit about how do we know what we know that's new. And most importantly, throughout, I want to try and emphasize why you should care about menhaden, this little fish that most people have never heard of. So hopefully by the end of the talk, you'll be in love with them as well. So I'll start with our first question, which is, what is forage? So when a fisheries scientist is talking about forage, we're talking about a particular place in the food chain, in the ecosystem, the aquatic ecosystem. So at the bottom of the food chain, we have these microscopic plants and animals, phytoplankton and zooplankton, and they are in great abundance, but they're really, really tiny. And big predators at the top of the food chain, like us, humans, as well as whales, sharks, large fish, and so on, they can't eat that plankton, right? We can't go around just slopping up zooplankton or phytoplankton. So in between, in the food chain, we have what we call forage animals. Some of them are fish, which of course then we call them forage fish. Some of them might be invertebrates like squid or shrimp. But these are smaller animals, typically, that are consuming these primary producers, the phytoplankton and zooplankton, and by consuming them, they turn them into their own biomass. And then the predators higher in the food chain eat those forage animals, and it moves that energy up through the food chain and circulates it through the ecosystem. Does that make sense? So these forage fish are really important parts of our aquatic ecosystems. It helps move that energy through the system. My favorite forage fish is Atlantic menhaden. You may hear them called bunker or pogie, depending on where on the East Coast you are. People will call them different things. They're a beautiful little fish. I shouldn't say little. People get the idea that they have to be small. They start off really small, but they can get to be quite large. So depending on how old they are, they can be much bigger than you might expect for a forage animal. But they're beautiful silver and they have a little beauty mark right behind their gills. And they form these really large, dense schools that when they come to the surface, which is most of the summertime when the water is pretty warm, they form these big schools that shine. If you see them from either from your boat or from the plane, they flash silver. And it's really beautiful. Like, you know, some of you must have seen them because they're absolutely gorgeous. Now, I'm talking about the mid-Atlantic, but tonight, but their range actually extends from way up in the Southern Maritimes, Maine, Gulf of Maine region, all the way down to about the middle of the east coast of Florida. The Chesapeake Bay is a really important area for them. I'll talk about that in a minute, but they do range across the entire East Coast pretty much, which means that they don't respect human boundaries. They don't care about state boundary lines. They don't care about state versus federal waters. And so that means that in order to effectively manage them, we need to have a different sort of way of handling that. And so the fishery management plan for Atlantic Menhaden is handled by the Atlantic States Marine Fisheries Commission. Before I came here to CBL, I worked for them as their senior stock assessment scientist for 8 years. And so they are an interstate compact, which is basically an agency somewhere between the state and the federal level, where the 13 Atlantic states got together and said, we need to manage these migratory fish because they don't respect state boundary lines. And if we ignore what's going on in Massachusetts and just manage the fish in Florida, we could be mismanaging them, right? We need to look at the whole population. And so the Atlantic States Marine Fisheries Commission, or ASMFC, as I might slip up and call them, manages Menhaden, and they do it at this coast-wide level. Now, each of the states may have additional regulations that are in place, but they can't be any looser than the strictest regulations that the commission sets for them. Does that make sense? All right, cool. So that's the commission. Now, I'm talking about fisheries management. There are actually 3 different types of fisheries that are important that are prosecuted for Atlantic Menhaden. On the East Coast, the largest fishery by volume is actually the Menhaden reduction fishery. We call it reduction. I'll explain that in a moment, but I'll just show you how it's done in this first slide here. It's basically what happens is, as I mentioned, these fish form very large schools, and during the spring, summer, fall, they can be seen near the surface of the water. And so they send out spotter pilots who can see these schools. They're kind of rare across the landscape, but when you see them, they're gigantic, right? And then they send out the boats to that location. And the big boat, which, let's see this, for folks online, I'll use the mouse here. Everybody can see that? So this big boat gets sent out to where the school is, and they deploy 2 smaller boats with what's called a purse seine. And they call it a purse seine because it's a net that says like a seine or a sieve. And it, like a purse, you know, when you pull up a drawstring on a really loose purse, that's exactly what happens. Once they've encircled the school, then they pull that purse up towards the boat, and it brings all the fish next to the boat where it's pumped out of that purse seine into the vessel hold. And so that's the purse seine fishery. The landings then are brought back to port. And it's called a reduction fishery because the animals are reduced down by kind of boiling them essentially into their component parts, where there's fish meal, which you can see on the left. Those are big piles of Menhaden fish meal. That's kind of the solids that are left after the reduction process. But the really valuable and important part of Menhaden is their oil. So they are extremely oily fish. You're never going to see Menhaden at the restaurant. There's no filet of Menhaden sandwiches anywhere for sale. The indigenous peoples of this region use them as fertilizer because even they wouldn't eat them. So they're really, really oily, too oily for most human palates. And so the value in them, though, is this oil. They're very high in oil, and those oils are high in omega-3 fatty acids. And so if you're into heart health and you take those omega-3 fatty acid pills from the pharmacy, take a look when you get home at the bottle and see, it might be Menhaden. It might be one of Menhaden's close relatives like herring or mackerel. But those pills are typically from one of these oily fish, including menhaden. And some of those oils are so refined and so high quality, they use them for things like racehorses as well. So there's all sorts of uses depending on the grade of the oil that's produced. So that's the reduction fishery, and that is the largest by volume. About 75% of the landings on the East Coast are for the taken by the reduction fishery, but they're not the only ones. There's also a very small recreational fishery, which is primarily folks who are going out charter fishing or maybe private boat fishing. They'll cast net to get some menhaden for bait before they go offshore to go get something else that they're targeting. But the second largest, that's actually just a tiny fraction though. The second largest fishery is the bait fishery. And so how many of you eat lobster? Yeah, if you've eaten lobster, then you've kind of in a way eaten menhaden probably, because they are a favorite bait of lobstermen for lobster pots. So the most favorite bait that lobster like are Atlantic herring, but herring aren't doing so well right now. And so they've switched largely over to Atlantic menhaden. And so there's about 25% of the landings on the East Coast go to bait, which primarily goes to the lobster pot fishery. So it's kind of the why we harvest menhaden. So this is a history here, just to give you some perspective of the landings of menhaden on the East Coast here. So on the left-hand side, we have the reduction landings, and those correspond with the blue bars. So what you can see is that the harvests were very, very high. In fact, I would say they're quite overfished in the 1950s. In that time, there was a decline, and then another bit of a boom again in the 80s, and that the landings have now declined to their historic lows. And part of the reason for that is that there used to be plants, reduction plants, all up and down the East Coast from Maine to Florida. Over time, with gentrification of the coast, shoreline development, and expansion of other oil products, there was a reduction in the number of reduction plants. I just realized that sounds funny, but it's true, such that now there's only one plant left on the East Coast in Reedville, Virginia. And so it only has a certain capacity, and so this is kind of the max capacity for that fishery. On the right-hand axis, so basically they ranged from about 700,000 metric tons down to our current level of about a little over 100,000 metric tons. If you look at the orange line, this is the bait and recreational fishery combined, but it's mostly the bait fishery. So this is that 25%. You can see that since we started collecting information on menhaden bait, they were using them for bait before that, but we weren't collecting the data on it. It varied over time, but it's been increasing in recent years. And that corresponds with the right-hand axis. So now we're at about 60,000 metric tons harvested for bait. So over time, there's been quite a bit of change in the fisheries on this species. So I will say that if you're into bay conservation and you follow the news and you follow social media, you may have heard a lot about menhaden. So I see some of you nodding your heads. Some of the information out there is spot on, but don't believe everything you hear. And some of it is contextual, right? So you need to be careful about hearing and interpreting the science that is shared online. So one of the questions I get asked most frequently is, what is the status of the stock? I hear they're in terrible shape, menhaden. The answer actually is that at the coastwide level, they're in pretty decent shape, probably close to the best they've been in a very, very long time. This is a graph of fishing mortality over time from the 50s when they were over-harvested through to today. And you can see here that they reached very high levels of fishing mortality in the 50s and then again in the 80s. But now we're at historic low levels. And in fact, at the end of the last assessment, they were below the target level. So you want the target to be low, right? If you're worried about fish conservation, that level was actually quite low. Because in large part, the reduction fishery has scaled back so much since the 50s and the 80s. And you'll notice here that I have the word ERP is in here. That stands for ecological reference points. So this target that they have for what the fishing mortality should be is based on not just the population and how sustainable the fishing is just for the Menhaden population. But it also takes into account whether there's enough forage for striped bass at their target levels. So this is actually it is the first in the United States fish that is managed using ecological reference points that account for more than just the single species that's being considered. It took 20 years to get here, but it's one of the first time fish, in particular, one of the first forage fish. There are a couple others in the world, but this is probably the first one where it's been really written into the fishery management plan. So we're pretty excited about that. Now, this is a graph of the fecundity. So in other words, this is the number of eggs produced by the spawning fish of Atlantic Menhaden over time. And so you can see here that there were quite a few of them in the 50s. It dropped down to pretty low levels by the 80s, but it's since bounced up, and we are currently above the target. Here, it's the opposite, right? You want the target to be a lot of eggs as opposed to low fishing mortality. Again, these are ERP, or ecological reference points. So these are ones that take into account, is there enough egg production in Menhaden to sustain the Menhaden population and the striped bass population at their target levels? So kind of neat. So the answer to the question, how are they doing? At the population level, they're doing just fine. Now, you may find that in particular bays or in particular parts of their range, they may be at different levels in different time periods, but across their range, they're doing quite well. Another soundbite you might hear is that Menhaden are going to clean up the bay. This is one I want to take a little bit of time to talk about. They are filter feeders, which is really cool, such that they bring water into their mouths and it goes through their gills and through their gill filaments, and they siphon out the plankton. When they're little guys, 1-year-olds, they eat a lot of phytoplankton, so microscopic plants. As they get to be 1-year-olds and older, those gills, the holes between them essentially, are so big that the phytoplankton fall through. They're pretty much only eating zooplankton. So here, I'll show you this really cool picture I found online. It shows a stained Menhaden body. And so what you can see here are these purple bits are their gill rakers, which see the striations there? Those are the filaments they're basically using to filter food out of the water. When they get to be 1-year-old and older, they're pretty much too big to filter out the phytoplankton that is causing these blooms that you see in the bay, and those blooms suck up a bunch of the oxygen and cause dead zones. So the story that's been going around is, well, if we just have more Menhaden, they'll eat more phytoplankton and that'll solve the problem. We'll clean up the bay. The problem is that most of the biomass of Menhaden is age 1 and older, and they can't eat the phytoplankton. And so there isn't enough biomass of Menhaden to clean up the bay, especially considering the fact that we are putting so many nutrients into our bay. We're the problem, and I'm afraid that Menhaden aren't going to be the answer. Our folks at our colleagues at Virginia Institute of Marine Science did the work on this most recently, and it pretty much debunked that idea. So if you want to clean up the bay, we're going to have to do other things instead of that. That's not to say that we shouldn't make sure there isn't enough Menhaden out there, but that's not going to be their main function. So that's kind of a really fast overview of why Menhaden are wonderful, who they are, and why they're cool. But what I want to get into is my research. And so to explain a little bit about what I'm doing and why, I need to give you a little bit of background on their life history and tell you kind of how they operate as a fish. So on the upper right-hand corner here, that's an adult Menhaden and a little larval Menhaden below them. The adults spawn in the coastal ocean. They don't spawn in the bay, they spawn out in the ocean, and the larvae then enter the estuaries. And they hang out there in what we call these nursery areas, basically. So as they're really little, young of year, 1-year-olds, basically, they hang out in the estuaries and grow and eat. And then as they become 1 to 2-year-olds, they start to migrate out of the estuaries into the coastal ocean, depending on their size and age. The larger, older animals then tend to migrate farther north. So as they get bigger and older, we find them farther and farther north, such that the largest animals we typically see up in southern New England. There are exceptions to that rule, as with everything in the ecosystem, but in general, that's the pattern. Now, when winter comes, the older animals that are out in the coastal ocean, most of them tend to go down farther south. They migrate south for the winter like a lot of animals. And the just so story that we scientists told ourselves for many, many years, including when I first started at the commission, I remember one of the older scientists telling me, you know, they all go south for the winter. They hang out off of Cape Hatteras, North Carolina. That there's no fish up in the mid-Atlantic, no Menhaden in the mid-Atlantic in the winter. So that was within my career, we were saying that. But I think I'll change your mind by the end of my slideshow here. So the question that I was set out to ask was, are they really all snowbirds? Are they really all going south for the winter? Because this was the story that we told ourselves for many, many years. So the first piece of information, or the first study, I should say, that was done here at CBL that started to answer the question was done by Kara Simpson, who was a graduate student under Dr. Hung Sheng Bi and Mike Wilberg. Now, she took a look at the available surveys that the federal NOAA folks do up out of the Northeast Fisheries Science Center in Massachusetts. They've been surveying for juvenile fish for many, many decades. So she went and grabbed their data set. And you can see here on the left, let's see if I got my pointer. These are the different regions in which of the ocean in which they sample for juvenile fish or larval fish. That corresponds to these regions on the right. So you can see the smaller numbers are the further south regions, and the higher numbers are the further north regions. So when she analyzed those data looking for evidence of Atlantic menhaden in those survey data, she found that there were juvenile menhaden, larval menhaden, all up and down the coast and in all months. So now we have to ask ourselves, how did those baby menhaden get all up and down the coast in all months if there were no adults there? So we started to scratch our heads and say, well, something might be going on here. So the next, the next study that we did was to look at a historic tagging study that was done in the 1960s. It was a very, very large, ambitious tagging study where NOAA folks out of the Southeast Fisheries Science Center in Beaufort tagged a lot of menhaden out at sea, all up and down the coast. You can see the different regions in which they were tagging them. And they put little magnetic tags inside them. And when they were fished and brought back to the port, as they were being processed into the facility, there was a big magnet, and it just caught the tagged fish, and they were able to figure out, looking at the tag number, where they were fished and when they were tagged, and what region they were from, so they could figure out how long they had survived and where they were from. And so Emily Liljestrand was a PhD student here at CBL. She worked under Dr. Mike Wilberg, and she found by reanalyzing this old tagging study that there were fish, menhaden, adult menhaden, caught in this survey, in this tagging study, in all months, in all of these regions. So that was like, okay, now we're getting somewhere. But then we had more naysayers. They said, well, that was in the 60s. Maybe things have changed. And I'm like, oh, really? Okay, well, maybe, because with global climate change, lots of things are changing. Animals are moving where they are. They're changing their migration patterns. That's possible. So to kind of figure it out and get the answer for real, we had to go out on the water. And so that's what I'm going to spend the rest of the talk telling you about, is our survey that we did in 2022 off the coast of New Jersey to look for menhaden in winter and finally answer this question of, are they all snowbirds or not? So the impetus for this survey was actually came from the fishermen. So the other reason that we started to get really curious about this question was because in 2014, Suddenly, in New Jersey, some of the fishermen who normally fish for squid in the summer and then herring and mackerel in the winter suddenly landed the entire New Jersey quota of Menhaden in one trip. And our eyes got very big, and that alerted the managers to, boy, something's going on up here. And we said, well, they said, we talked to them and they said, well, we see them all the time. You just never asked us. Oh, well, why are you suddenly harvesting them? Well, again, herring is normally their target. Mackerel is their target species in the winter, but those populations have gone down and the quotas have gone down. So they were looking for something else, and this is a good bait species. So suddenly they were targeting them when they weren't before, but they said they were always there. We just never asked them. Very interesting. So they said, well, we caught the whole quota in one day. There's plenty more out there. Can we have more quota? And the managers reasonably said, well, we don't know how many fish are out there, so we don't know how many are safe for you to be catching. Why don't we figure it out? And so we all got together and did this survey to try and figure out how many fish, Menhaden, I should say, are off the coast of New Jersey in winter. So this was a cooperative research project. And by that, I mean, we had a wide range of scientists and fishermen working together on a research project. And you'll see why we needed the fishermen in a moment. So we had academic and private scientists involved. Chesapeake Biological Lab was the lead, and Dr. Dong Liang in the back there was my co-PI on this project here at CBL. But we also were working very closely with folks at the Virginia Institute of Marine Science, who are outstanding survey biologists and really provided a lot of the on the ground, I should say on the water work on this survey. And then we had acoustic experts, sonar experts from Normando. We had industry collaborators from Lund's Fisheries, who are a bait dealer in New Jersey out of Cape May, and they helped us with weighing the Menhaden that we sampled when they came to port. I'll tell you why that was important in a minute. And we used the fishing vessel, the Durston. I'll describe that too in a second. And we had two amazing captains who have a lot of knowledge about the water, about the fish, and about that general region in winter, without whom we couldn't have done this survey. We had several federal and state partners as well. The NOAA Northeast and Southeast Fisheries Science Centers were involved, as well as New Jersey Department of Environmental Protection. And the Atlantic States Marine Fisheries Commission, which is responsible for managing Menhaden, made sure that we had adequate quota in order to make sure we were legal and following the fishery management plan and didn't go over when we were sampling those fish. So that was greatly appreciated. So this project was funded by NOAA through the Saltonstall Kennedy Competitive Grants Program. And the goal of this program is to do science that supports sustainable fisheries, to make sure that the fishing opportunities and fisheries that are out there in the United States are sustainable. So our goal for this particular project was to conduct a sonar survey. And does everyone understand? Sonar is when you basically go out with a boat, you attach this unit that sends sound out into the water, and the sound waves ping back and you sense those. Usually you're getting mostly the sea floor, but if there's a school of fish underneath, you can see that school of fish, and you can get an estimate of the species composition sometimes. You can get an estimate of the biomass of that school from that sound data. So sonar is really important tool for surveying fish. So we wanted to go out and do the sonar survey to try and figure out how many Atlantic Menhaden were overwintering off of New Jersey, to see whether or not this winter bait fishery was sustainable or not. So we set out to try and estimate the biomass within that region off of New Jersey, as well as to try and get a handle on what the age and size composition of those fish are. Are they young fish? Are they old fish? We didn't know. What's the sex ratio? Are they spawning animals or are they immature animals? We really had no idea. And then we were also interested in evaluating whether the sonar that's on the boat on a fishing vessel can be used for these kinds of scientific surveys. So this is the study area. To orient you, this is New Jersey right here, and Cape May located, that was our base of operations. This is the Delaware border. So we were surveying from about 15 to 50 miles offshore. And we were basically concentrating in this area from southern Hudson Canyon down to the Delaware border. We ran 6 transect lines where we told the fishermen, you start here and you go there, and we will see how many fish we encounter along the way. There we go. This survey was done, as I mentioned, using a fishing vessel, the FU Durston. This is a very large vessel. It's 160 feet. It's a midwater trawler, meaning that they use a trawl net to catch fish in the middle of the water, as opposed to a bottom trawl, which is what a lot of cod or other ground fish are fished, the method that's used to fish for them. We had 2 very experienced captains, as well as the VIM survey crew on board, which was amazing. And this vessel was so large, we had the ability to set up an entire lab on board to do all of the sampling of the fish with really advanced, really cool boards where you just set the fish down and it would measure the fish and it would weigh the fish and it went directly into the computer. You didn't have to do any paper and mess up by writing the wrong numbers. And yeah, it was all absolutely amazing and technologically, the wizardry was wonderful. So this vessel was really important because it's equipped with some of the most advanced industry grade sonar that's out there on the market. It has both a down sounder. So if you've ever used like a hummingbird on your boat, it's got a sonar that's pinging down underneath the vessel, as well as one that's kind of like this green cone. It's also sending, has an omnidirectional sonar that's looking out in front of the boat. And so that allowed us to not only get pictures and data on the biomass of the fish that we're going over while we're running those transects, it also let us broaden our search area by looking out in front of the boat to see where those schools were. And I'll show you how that works in a moment. So this is the survey that Dr. Liang helped us design, and he simulation tested this for us. So basically with a normal acoustic survey, you would run your transect by just using the down sounder. And that means you can only really sense fish that are under the boat. But with Menhaden, they're not evenly distributed across the sea, right? They're in these big dense schools, but those schools are pretty rare across the oceanscape. And so what we were able to do was broaden our search area by saying, okay, you look out using the omnidirectional sonar way out ahead, and this thing is so advanced, it could get over 3 kilometers of a breadth. Yeah, about 1.5 kilometers each side of the boat. You could sense whether there was a Menhaden school out there or not. And if it did, and if we were able to see it, it was in that path, we would deviate from the transect, go take the scientific information on it, and then go back to the transect and keep running. If we saw it, but it wasn't in that swath, we noted its presence, but we didn't include it in the estimation of biomass because that would bias it upwards, right? So thanks to Dr. Liang, we were able to develop this very Menhaden specific survey design that allowed us to get out there and effectively survey for these animals. This is a picture of the wheelhouse. It's the size of this room, pretty much. It's just to give you an idea of all of the acoustic and mapping that's required to operate this kind of a vessel and a survey. It was pretty impressive. So we were able to get images like this one in the upper left-hand corner of gigantic Menhaden schools. These are absolutely, if you ever look at sonar images on your boat and have seen schools of fish, you can imagine this is in 50 fathoms of water. This is really a lot of Menhaden. But these images are pretty messy. And so our acoustic expert was able to take them and use a bunch of filtering to come up with a final image that allowed us to get a much better, clearer picture of the biomass underneath the boat, which was pretty exciting. We were also able to check whether or not our estimation of biomass from those images was realistic or not by actually capturing entire schools of Menhaden. And that was one of the advantages of using this very large vessel. People said, oh, with schools like this, you analyzing this kind of acoustic data, the sonar data, it's not terribly reliable. You're gonna have to prove to us that you're able to do this accurately. And we said, okay, well, we'll just collect the whole school and then weigh it out at the dock and compare it with our estimates from the sonar data. We only did that for a subsample of them, but it allowed us then to individually place schools in different holes in the vessel and then pump them out. This is our captain here and our chief scientist overseeing the pump out of the individual schools into the facility where Lund's Fisheries was able to weigh them individually. And this is me standing next to all the fish that we caught on our first 2 transects, 240,000 pounds of Menhaden packaged up in boxes. So yeah, we were able to show, I'll show you in a minute, that we weren't too far off the mark with our data analysis by actually harvesting and weighing out a sample of those schools of fish. So hopefully in the future, we don't have to do that. The idea being that we've proven that hopefully. But when we did harvest schools in Menhaden, we took full advantage of that opportunity and subsampled individual fish to get information on their biology. How big are they? What sex are they? Whether they were mature or not. And we also collected both scales and otoliths, which are ear bones, which can be used to age the fish, kind of like on a tree where you count the rings. You can do the same thing with Menhaden scales and otoliths. So we were able to do that as well. We also took advantage of the opportunity to collect information on water conditions while we were out there at sea. So we dropped these hydrosondes into the water at regular points along the survey, and we're able to get from the surface to the bottom temperature, salinity, and dissolved oxygen information, as well as at all the locations where we spotted Menhaden. So lots of cool information. We ran the survey in February of 2022. We took additional samples, biological samples on board while they were continuing fishing throughout the remainder of the fishing season, which was really nice. The crew, the captains, and the folks at Lunds were really excited to do all this science. And so they put a lot of extra effort in than they ever committed to at the very beginning. And we were grateful for that. As a result, we can say Menhaden are not all snowbirds. We caught a lot of Menhaden. We observed a lot of Menhaden schools, over 100 schools we collected sonar data on, and 5 of them we actually trawled up and sampled and then weighed out individually at port. We collected lots of water condition data. This map on the right shows our study area and our transects, and the black dots are all the locations of all the Menhaden schools that we saw. And the red triangles are the Menhaden schools that we saw after the survey while we were just on board collecting biological information while the fishermen continued fishing. So a lot of Menhaden. We collected over 4,000 length and weight, paired length and weights for individual fish, as well as over 300 fish had full workups of what were they, sex, weight, maturity, and age. Very important. This is really unique, and this is why. So when we get most of our data for Menhaden for science and management from the reduction fishery, which operates primarily in the Chesapeake Bay region, and these are mostly age 1 and 2. This survey, we were focusing on bigger, older Menhaden found farther up north. And you can see here that most of the fish that we encounter in the reduction fishery are about 250 millimeters. Here you can see most of our fish were much, much larger. As well as the reduction fishery is primarily harvesting ages 1 and 2, but we were seeing mostly animals aged 3 to 5, some as high as 6 possibly. And the females were larger than the males, which is kind of fun. That's normal for fish, but also it was cool to see in our actual data for Menhaden. So this was a unique opportunity to collect a lot of information on a part of the population that we almost never see in the normal fishing operations. We were happy when we started to compare our data with some of the data that's used in the assessment. This is just very quickly the fork length and age of Menhaden. In the assessment are the black dots and the black lines. The red is our survey. And the blue is another survey that's done along the East Coast from about April through September. And so you can see these kind of all line up. And so we were really excited to see that our data were lining up, and that's great. And then that's for fork length, but then we plotted weight at age. And we saw that the weighted age that goes into the stock assessment looks quite a bit different than the 2 surveys, our survey and the other coastal survey, the NEMAP survey. So if you compare the average of our survey data, the purple line, with the black line, which is what goes into the assessment, you can see these are very different weights at age. Once you get above age 3, these 2 lines diverge. And this could be a big issue for the assessment if this turns out to be a problem, because we use weighted age to determine the biomass. And we use biomass to figure out what the landings quota should be. So that's kind of a big deal. So we just got a grant last week, actually, to address this issue, hopefully in time for the next assessment. Very briefly, we found that it was about 50-50 sex ratio. But the really interesting and important thing for management is that most of the fish that we encountered, at least in 2022, were mature, but they weren't actively spawning. So fish have a tendency, some species, to do these big spawning aggregations where they have one place they all go and they do their spawning. And if you fish on them while they're doing that spawning thing, that can be really bad because they're kind of sitting ducks, if you will. When I said we were gonna go out in winter, where these bigger, older fish are, people were concerned that we were gonna be bothering these spawning aggregations. Turns out that's not the case. So that's really good news. It kind of makes sense with some of the other research that had shown that they're spawning throughout the year and all up and down the coast. But this is more confirmation that this isn't a spawning aggregation, but they are a lot of mature fish. And then one of the other really cool things we found was that, remember how I said we harvested the whole school for 5 of those schools? If you look at the red bars, these are the weights of those schools that we got from bringing them back to port. And the blue bars are estimates from the sonar images. So even though they differ a little bit, in most cases, they were pretty close. And so that gives us hope that even though it required a bit more data processing, and was a little bit trickier to do using industry grade sonar, we think that this is an opportunity. We could use this in the future. It's much cheaper and it's already on the boat. And so that means there's less problems with having to attach sonar to somebody else's boat. This is a really good opportunity for future research, especially for Menhaden, because it's hard enough to get Menhaden surveys going to begin with. And so I want to finish with just some results here that Dr. Liang did for us here, where he took our data and ran a series of models that estimated what, from our surveys and our encounters of Menhaden, what the total population is in that area off of New Jersey. And we estimated that the total biomass of Menhaden in the region was between 17 and about 24 million pounds, which sounds like a lot. But if you look at the estimate down here of the total coastwide adult Menhaden biomass along the entire East Coast, it's over 3.5 million metric tons. So it's actually a tiny proportion of the population is living off of New Jersey in winter, but it's an important part of the population, and they don't all go south for the winter. So that was good to know. The other interesting thing from a management perspective is that the typical New Jersey winter trawl quota, which is what those bait fishermen were operating under, is about 680 metric tons. So that means that they're only harvesting about 6 to 9% of the population when they take that. And honestly, that's an overestimate because they don't just fish off of New Jersey. They fish between Maryland and as far north as Rhode Island, Massachusetts. So they're having a negligible effect on the total population, most likely. So that's good news, but we had to check to make sure. And just lastly, I just want to show you some cool images that I thought you would like that we were able to get while we were out there. This isn't formal data, we're not going to publish it, but I thought you would like it. This is a picture of one of the Menhaden schools we found. Guess what those little guys are? Anyone want to hazard a guess? Yeah.
Are they dolphins?
Oh, no, they're actually a little smaller than that, but we think they're probably striped bass.
Oh.
Yeah, fishing themselves on some Menhaden. Yeah, pretty cool, right? And then the other thing is that Menhaden do this crazy behavior where during the day in the winter, they form these big schools, right, near the bottom, which you can see on the top here. But at night, that's when they kind of wake up and go to feed on the surface. And so that giant Menhaden school disperses and they go up as individuals to the surface. So what we asked the vessel and the survey crew to do was just hang out over one of the schools as they finished for the evening and just keep the sonar going. And we were able to see them just kind of dispersing off into the really cool stuff, fun biology. So in conclusion, I just want to wrap up with a few take-home messages. The first is that we have finally fishery independent confirmation that these aren't all snowbirds. There are overwintering Atlantic Menhaden in the mid-Atlantic. That's a huge career victory for me. There also is, of course, then a small portion of the population that's overwintering off New Jersey, but it's as far north, we think, as Massachusetts, possibly farther north. The New Jersey winter trawl quota is likely sustainable, which is good news for management. And the big take home for all of Menhaden, I think, is that we can't use a traditional acoustic design. We have to do something different for Menhaden if we're going to successfully survey for them. If we had just run with that down sounder that was pinging under the boat, we wouldn't have seen any Menhaden. Zero. but they were all, there's 7 to 24,000, or excuse me, million pounds of them out there likely. And so that this just says that we really need to do something different for these schooling fish that are, that don't behave like all the other fish. And so this could be applied not just to menhaden, but to herring, possibly mackerel and other species. And I'll leave you with just a plea that if you take nothing else from this talk tonight, I hope you will remember one thing, and that is that many of our fish that are in the Chesapeake Bay, including menhaden, are migratory. They don't spend their entire lives in the Chesapeake Bay. Maybe a very important part of their life is in the Bay, but they don't spend their entire lives in the Bay. And so to accurately assess them, to properly manage and conserve them, we need to be looking at their whole population, wherever it may range. I feel like it's really important that folks in the Bay who are so passionate about our menhaden and other fish species, that they continue to be passionate, but we need to be passionate about the whole range, because if something happens up in southern New England or New Jersey, that could impact Chesapeake Bay and vice versa. So all of our fish from Atlantic menhaden, rockfish, bluefish, summer flounder, weakfish, you name it. There are so many fish in the Chesapeake Bay that people love that are migratory. So really, if you're thinking about the Bay, keep thinking about it, keep thinking about Bay conservation, but remember that many of these fish are impacted by stuff that's going on out in the coastal ocean as well. So if we care about our predators, we care about our striped bass, we care about menhaden, we need to be thinking coast-wide, as well as concentrating on the Bay. And with that, I will stop and see if you have any questions. Thank you.
Thank you so much. I've learned so much. So I'll take a couple questions from the audience here first, and then I'll turn it over to Dr. Miller if there's any questions from those of up in the Zoom land. So any questions here? Oh,
I was going to ask, are menhaden the only fish that present like that in the big schools on the southern screen? Is that how you identify them primarily?
Yeah, so the menhaden here, I'll actually pull back here and show one of them just to give, just so you can see what I'm talking about. Let's go back here. If you look at this image, like school 49 there, see how dense and red it is? That's menhaden. There is nothing else out there that looks like that.
The density.
Density, yeah, and it just beams like bright red. If you see herring, they will look like that, but they don't go as dense as that. And so they'll look more patchy and they won't be as bright red. Mackerel don't have the same swim bladder thing that allows the sound to bounce off of it the same way. And so they look completely different. I wasn't convinced, and the other scientists weren't necessarily convinced that the fishermen could tell the difference that easily until our chief scientist went out on the water and he saw both herring and he's like, oh, okay, I believe you now. As well as in all, I'm trying to remember in total how many thousands of pounds of menhaden we harvested from those 5 schools, but we caught, in the subsampling that we did, we caught no mackerel, no herring, a couple striped bass. I think there were a total of maybe 10 other species, like 10 individuals of other species, very, very concentrated, all menhaden. Does that answer your question? Cool. Question? Yeah.
You mentioned that when you captured the schools and you were able to get actuals versus your estimate, you said you were very close. How close were you, and was the delta consistent across schools?
No, it wasn't consistent. So some of them were where did I have it? So you can see some of them were similar, like school 10. Some of them, our trawls caught more than we estimated from the sonar. Sometimes it was the other way around, like school 13. Part of the reason for that, and we don't expect them to line up exactly, because in large part because these are giant schools of fish, and sometimes when you lay the net down, part of the school will just peel off. So we may have passed over them, gotten the sonar data, turned around, set the net, started to harvest them, and some of them might have already peeled off. I expected them all to be underestimates, the trawl catch, because of that behavior, but sometimes it looks like we were off, so there's other things going on. One of the other challenges we had was that where did it go? Where's that image again? This is not your normal image. When we go to analyze sonar data, they're usually more like a ball or an oval. And so we use the equation for the volume of an ellipse, basically, that kind of oblong shape. This doesn't look anything like that. So we used a dome, the volume of a dome. So we had to do different stuff than people normally do when they analyze sonar data.
Thanks.
Yeah.
Hey, Dr. Miller, any questions from the Zoom audience?
Not at the moment, but I'll interrupt if we get some. So those on Zoom, please type your questions into the chat if you have any.
Thank you. Any more questions from the audience here?
I wanted to ask, like Deltaville at Omega or whatever it is now, the Canadian firm that does the rendering there.
Yeah.
Is that mostly juvenile fish or, I mean, if they're in the bay, are any mature fish brought in for rendering at that plant, or is that just a local fishery? So,
Right. So most of the catch are ages 1 and 2. They occasionally do fish as far north as New Jersey. There's very few sets up there, but they do that sometimes, typically more in the fall. They'll do that. And so then they're getting ages probably 3 and 4 sometimes, but it's honestly, if you look at the composition of the catch, it's almost all ages 1 and 2. In a few years, it might occasionally over the 70 years of data we have, they'll get a little bit more age 3s or so, but it's mostly ages 1 and 2. Yeah. So they're not larvae, they're not the young year that they're catching. That's too small. It's ages 1 and 2.
Okay, any final questions? Okay, well, please join me in thanking Dr. And thank you all for coming out again. And just a reminder, next Tuesday, 7 o'clock, same time, same place. If you're interested in emerging contaminants and what the Chesapeake Biological Lab plans to do to look at those, please join me next week for Dr. Michael Bonzier and Dr. Andrew Hayes' seminar. Thank you, everybody.
Her daughter came here to visit CBL and you talked to the little guy. He's going to speak at college. Okay, I'm not going to. Yes, they are.